So today I'm going to talk about peripheral B cells, sort of one of our last pieces of putting together all the parts of the immune system before we can actually start to move into some of the more clinical and applied stuff. I usually think of today, the peripheral B cell stuff today, NK on Friday, and then the first peripheral tolerance lecture as, to me, the end of like basic immunology, and after that is clinical. I think sometimes students think that the first peripheral tolerance is also clinical, so I think we might read that one a little differently of where we are on that line, but we're moving towards those clinical topics. One of the sort of things that's a little bit of a trick in thinking about peripheral B cell responses, this is actually why I sort of have not given you the paper yet for next week, or the assignment for next week, is that I would say, at least for me personally, but I also think for the field in general, this is an area where we have learned some of the most new immunology during SARS-CoV-2 because of some really cool studies that people have done on peripheral B cell responses, and some of the things we're going to talk about today when applied to both SARS-CoV-2 infection and vaccines. And so trying to decide how I want to take like all of the coolness that has been learned in the past 4 years and get it to you in like a day is where I'm struggling right now. So basically, to give you a little bit of orientation to where we are right now, we're thinking right now about B cells in the periphery. So remember way back, we talked about B cell development, where B cells were in the bone marrow, and then we're doing development, and then we talked about the idea that they could leave, go to the periphery, particularly secondary lymphoid organs like lymph node or spleen, as is shown here, the pink areas, and actually respond to antigen. But we never actually talked about the details of that response. You'll see why that is as we talk today. And so today we're really going to be thinking about sort of what's going on in these pink areas with B cells. So, circulation of the B cells, I'm not going to say a ton about that. It follows the same trafficking rules we saw before. Activation of the B cells, expansion of the B cells, differentiation of the B cells. So similar kinds of steps to what we saw with T cells in the periphery, but now we're going to see the B cell version. As is implied here, the secondary lymphoid organs are going to be super important, just like they were super important for activating peripheral T cell responses. I actually moved some slides around last night, so this is one of my slides I moved, so let's hope I like this location for it. So that secondary lymphoid organ is going to be particularly key, and the big reason why those secondary lymphoid organs are going to be key is that those secondary lymphoid organs are going to be collecting antigen, T cells, and B cells. So we will have our lymphocyte, whether it's a B cell or a T cell, traffic into that lymph node through an HEV, and then go to the B cell area or the T cell area or wherever is appropriate for it. And you can see here a naive B cell coming in and going to a B cell area, a naive T cell coming in and going to a T cell area. So all of that happening. You can see a dendritic cell that may have come in as well, hanging out in that T cell area. And the other thing that's not drawn super well on this slide is that we also have, if you recall, free antigen coming in through the lymphatics. So if we have just random proteins, carbohydrates, lipids, whatever, that are getting caught in the lymphatic fluid when it's getting pulled in, or random microbes, that can also be a free antigen that's also coming into the secondary lymphoid organ. And so on that previous slide, I mentioned that the secondary lymphoid organ was collecting up the antigen That could be antigen that's on a dendritic cell. That could be free antigen that you saw on the next slide. T cells and B cells. And the collection of all of those things in the secondary lymphoid organ is going to be particularly important. This slide also mentions something else about that free antigen. That free antigen doesn't often stay free when it goes into the secondary lymphoid organ. So it doesn't always stay floating around free by itself. Most of the time, that free antigen ends up getting bound to a particular cell type. These cells are perhaps the most annoying cell in all of immunology in terms of naming. These cells are called follicular dendritic cells, or FDCs. You can see that at the top. The reason why those cells are called follicular dendritic cells, or FDCs, is because they are in a part of the secondary lymphoid organ called the follicle. So they're the follicular dendritic cells. But there's a problem. Follicular dendritic cells are not dendritic cells. They are not at all the type of dendritic cells that we've talked about before. They are a totally unique, totally separate, totally different kind of cell, only found in the follicle of the lymph node, doing something completely different than the other dendritic cells you learned about earlier in the semester. In fact, regular dendritic cells that we learned about earlier in the semester, those are immune cells that develop from the bone marrow, like from monocytes. Follicular dendritic cells don't come from monocytes. They're not immune cells. They don't come out of bone marrow. They are completely and utterly not dendritic cells. The super annoying thing is that the people who first found them saw them under a microscope and saw what they looked like. And in fact, this is what a follicular dendritic cell kind of looks like. And what does it look like? A dendritic cell. So they just named it a follicular dendritic cell because they're like, oh, this looks like it's a dendritic cell living in the follicle. It's totally not a dendritic cell. The way I often like to think about follicular dendritic cells, and we'll see much more about this here. You can see a follicular dendritic cell. Is that follicular dendritic cells have tons of receptors on their surface so that they can stick to antigens. I think of follicular dendritic cells as these little sticky patches in the lymph node. They have complement receptors, which is what's shown here, so that if an antigen has complement on it, that will stick that antigen to a follicular dendritic cell. Follicular dendritic cells have Fc receptors. They have tons and tons and tons and tons and tons of receptors. And so I also think of them sometimes as like fly paper. You know that like tape paper that you hang up, and if there's flies around, they just fly into it and get stuck? That's how I think of a follicular dendritic cell. It's just this really little, it's this sticky cell. Oops. So that if whatever antigen comes into the lymph node, it just gets stuck on the follicular dendritic cell. So you can think of the follicular dendritic cell as just kind of this sticky patch where all the antigens are going to end up getting collected, the free antigens. Don't think about them as they're not going to do any phagocytosis. Don't think about internalization. Don't think about presentation on MHC, class 1 or class 2. They are literally just getting antigens stuck to them. And that antigen is not going inside the cell. It's not getting processed, not going on MHC. It's just getting stuck like it was a sticky piece of fly paper. The one nice thing, of course, is that follicular dendritic cell has a really big surface area because of how all of these like stretchy pieces. So it can get a lot of antigen covered too. It can be just like covered with all the antigens that are going to come in and stick to it. And so you can sort of imagine, instead of that free antigen coming into the lymph node and like floating around, it all just gets stuck on follicular dendritic cells. But of course, our goal today is thinking about activating B cells. When we think about activating B cells, there are two big classes of B cell activation that happen. Sometimes we classify antigens in terms of which way they activate B cells. In big picture, we have some antigens that are known as T-dependent antigens. So they can only turn on a B cell if there's also a T cell involved. That's why we had to talk about T cells before we could talk about any of this. There are some responses and some antigens that are T cell independent responses. So there are some situations where a B cell can get turned on without a T cell in a T cell independent response. What I want you to notice here, and I'm going to say so much more about this as we go today, is if you look at the B cells, or in green on this slide, if you look at this B cell interacting with a T cell to become activated in a T-dependent response, you can see that there are a lot of signals coming into that B cell, like a 1 and a 2 and a 3, kind of reminding you of what you saw with T cells, getting signals 1, 2, and 3. What you can see with a T-independent antigen is that in some cases you can get sort of a second signal, maybe through a pattern recognition receptor, like a toll-like receptor. This kind of antigen can actually also get a second kind of signal. It's just not totally shown up here. So the basic idea is kind of how we're getting those second activating signals for the B cell. But there's one other big thing to know about T-dependent versus T-independent antigens. That's listed here at the bottom. This table does give you some ideas of the chemistry of these different antigens. I don't care so much about that. But what I do care about is if you look at our T-dependent antigen, you can see that when we have a T-dependent antigen, we get class switching. Remember, we talked about class switching in antibodies. If we have a T-independent antigen, we don't have class switching. If we have a T-dependent antigen, we get affinity maturation. We get that situation where the antibody is going to start binding to antigen better. We get that improvement. That doesn't happen with T-independent antigens. If we think about memory responses, we see that with T-dependent antigens and not T-independent antigens. And so most of the B cell responses that we think about and that we talk about are really part of those T cell dependent B cell responses. And so by and large, everything else I'm going to tell you today and sort of going forward about B cells is going to be about T cell dependent B cell responses. So I want you to notice here, yeah, there are some situations where we can have B cells that are getting turned on in a T-independent fashion. But big picture, we're thinking about T-dependent responses. And that's because they do the fun stuff like class switching, affinity maturation, and giving us good immunologic memory responses. You can see this here as well. Here is a B cell that's interacting with a T cell. This is a T-dependent response. And then when that happens, the B cell is going to proliferate, just like you saw with T cell activation. We're going to get memory cells. We're going to get effector cells. We're going to get class switch. We're going to get affinity maturation. And so all of these pieces that are really key for a good B cell antibody response require the T cells to be involved as well, require us to have a T-dependent response. In general, if we are able to measure that antibody response, there probably had to be clonal expansion. If I can actually measure that you have antibodies against something, you probably had to go through this process, and so it was probably a T-dependent response. Most frequently, we measure IgG antibodies. IgG antibody production requires class switch. So if we're ever talking about IgG, there had to be a T-dependent response. So T-dependent responses are what we're going to be thinking about in this process. And that's why I didn't tell you about B-cell activation right after we talked about B-cell development, because this process requires T-cells to do something too. So we needed the T-cells before we could see B-cell activation. And so I'm going to talk through the details of this process on the next few slides. So the first thing that is going to happen when that B cell is in the lymph node is the B cell is going to encounter antigen. And so our B cell is going to encounter or see its antigen via the B cell receptor. And so you can see that happening here. So maybe in this case, you can imagine a little piece of antigen floating around. We'll go with that for right now. Here's a little piece of antigen. It was floating around. See, look, it was floating. And that it bound to the B cell receptor. The B cell, just like we talked about when we talked about MHC class 2 presentation, will internalize whatever has bound to its B cell receptor by receptor-mediated endocytosis. And so you can see that internalization of the antigen here. And once that antigen is internalized, that antigen will get degraded and will get presented on MHC class 2. So this is exactly what we saw when we talked about B cells as antigen-presenting cells. And this is the first thing that a B cell will do when it sees antigen in the lymph node. And that will start to activate that B cell. So you can see that that B cell is now going to start making class 2. That B cell is also probably going to make additional proteins like our co-stimulatory molecules, B7, otherwise known as CD80 and CD86. So it's sort of like we're going to start to wake up the B cell. We're going to start to turn that B cell on. But the B cell has not yet gotten sort of additional, what we'll call, say, signal 2 or signal 3 to be fully activated yet. It's just getting this signal 1. There's one other thing that this B cell does when this B cell starts this activation process. And the other thing that the B cell will do is that the B cell will move. So on the left, you see kind of the beginning of what's going on in the lymph node. You can see the B cells come in through the HEV, through the normal trafficking process we talked about. And when they do that, they go to the B cell zone. Helpfully named because it's where the B cells go. T cells also are coming in through that HEV, and T cells are going to go to the T cell zone. And that's where they're going to interact with actual dendritic cells and get activated through all the steps we have been talking about. The signal 1, signal 2. We're going to, as you can see here, see that T cell proliferate. So all of these T cell activation steps that we've previously talked about are happening at this time. At the same time, our B cell is getting activated in the B cell zone. Perhaps it is seeing some antigen from a follicular dendritic cell. As we activate that B cell, like I said, there's this one other thing that's going to happen, and that is that B cell is going to move. That B cell is not going to stay in the B cell zone right next to the follicular dendritic cell anymore. Instead, the B cell is going to move right to the border between the B cell zone and the T cell zone. And so you can see here are these B cells. They've started to get activated, and they have moved to this border region between the B cell zone and the T cell zone. if you had to guess why the B cell moves to that border region, why might you guess the B cell moves to the border region? Yeah, Jonah. So that it can interact with the T cell, exactly. So this B cell now needs to interact with the T cell, so it's going to kind of move to the border of the T cell zone. Remember that the B cell is now presenting some antigen on MHC class 2. What type of T cell interacts with antigen presented on MHC class 2? CD4. And so this B cell that has gotten a signal through its B cell receptor is now going to present antigen on MHC class 2 and interact with a T cell. That T cell is going to provide some cytokines to the B cell. So you can see that the T cell is providing cytokines. And the T cell is also going to have a protein on its surface called CD40 ligand, which is going to interact with a protein called CD40 on the B cell surface. That's basically giving the B cell signal 2, as well as those cytokines giving signal 3. And so you can think of CD40 on a B cell as being sort of analogous to CD28 that you saw on the T cell. They're both the things that receive signal 2. In that case, CD40 ligand, which is on the T cell here, is analogous to B7 that was on the antigen presenting cell. And so it's the protein that is providing the signal 2. And the only way that a T cell starts to make CD40 ligand is because it has been activated. So we're not going to have a naive T cell doing this process. Remember, we also didn't have naive T cells making cytokines. This T cell had to have done that activation step in the T cell zone previously before it is going to interact with the B cell. Once the B cell gets that signal 2 via CD40, as well as that signal 3 via a cytokine, now we're going to start to see something, the entry into something called the germinal center, which I'll show you more about on future slides. Again, the bottom of this shows that, yes, technically there are some things that can happen besides the germinal center. I'm not going to talk about them. I'm only going to talk about the germinal center. You can see this same process happening over here as well. Here's that B cell heading to the germinal center and now doing things like class switch, affinity maturation, as well as becoming an effector cell. Yes, Brenna. Yep. So I'm looking ahead at where a couple of my slides are. It looks like that also shows up in 2 slides. So can I repeat it there? Okay. Yes, Juliana.
You may have said this, but I just want to clarify the B cell zone.
So the B cell zone is a part of the lymph node or a part of the spleen. Sometimes we also refer to, this is going to help me, as a follicle. Okay? Remember we saw the follicular dendritic cells? So sometimes the B cell zone we'll sort of say is the same as the follicle, at least for our purposes. Okay? So it's a part of the lymph node or a part of the spleen. This T cell help that this T cell is providing, because that's really what this is doing. This T cell is helping the B cell become fully activated by providing cytokines and providing CD40 ligand. This is happening kind of right at that border of the T cell zone and the B cell zone. So Juliana, what might we call that area? The follicle, right? The T cell is doing help in the follicle. What might you call this T cell? Because it's actually not any old helper T cell that does this. It's a special kind of helper T cell. What might you call that special kind of helper T cell? Anyone? A follicular helper T cell, who you've already seen. So this is our T follicular helper cell. It's providing help to B cells in the follicle. And so it had to get polarized by its first interaction with antigen, just like we saw before. That will push that cell to become a T follicular helper cell. And then that T follicular helper cell will make cytokines that will help that cell become a T follicular helper cell. And so I guess just to go back for a second, here's my naive T cell. It's in the dendritic cell. It's getting told, grow up and be a T follicular helper. While that is happening, the B cell is seeing antigen over here, and then they're going to come together when they've both done some stuff. The B cell still has more stuff to do. The T cell does not. Well, I guess its only stuff is to help the B cell. Yep, Brian. It happens in all secondary lymphoid organs. So I use the lymph node as sort of my example secondary lymphoid organ, but this is happening in any secondary lymphoid organ. And so, Brenna, hopefully this will help. So here you can see, here's my B cell. It's internalizing the antigen, degrading that antigen, and presenting that antigen to the T follicular helper or the TFH cell. Now the TFH cell makes CD40 ligand. Well, it actually made it before. Our TFH cell provides CD40 ligand. Our B cell has CD40. That gives the B cell signal 2. And so in a T cell case, this protein on the surface of the T cell that was receiving signal 2 was CD28. So in this case, CD40 on a B cell is analogous to CD28 on a T cell. CD40 ligand on the T follicular helper cell is analogous to B7 on our antigen presenting cell. I know. But does that make sense?
Yeah, so CD40 on the B cell,
Mm-hmm.
CD40 ligand on T cell, B7.
Correct. And so you can see this happening here. One other thing that I want to point out that is shown on this slide, and it's something that we can talk about more perhaps at a later date, is that if you look at this antigen that is binding to the B cell receptor, what they're trying to depict in this figure is that this antigen has sort of multiple epitopes or multiple domains. It's got a blue part and a red part, right? Which epitope binds to the B cell receptor? Blue. And what you can notice is that when the antigen gets internalized, it gets all broken up. I don't know why there's yellow dots, but there's yellow dots too. There's some blue pieces and some red pieces that get made. It doesn't have to be the exact same epitope that gets presented to the T cell. So in this case, you can see it's a different portion of this antigen that was bound by the B cell receptor and that is bound by the MHC class 2. It could be the same, but it doesn't have to be. As long as there is something that can get presented by MHC class 2, that's a part of the thing that got internalized, this whole process can work. I say that because there are some situations where we actually use this in vaccines, where we actually are basically linking things together so that one thing binds the B cell receptor and one thing binds the T cell receptor. It's a pretty common phenomenon actually in a bunch of vaccine settings. It's known as linked recognition because the idea is the two things have to be linked. They have to be physically bound to each other so that they're both internalized at the same time. And it just so happens that this slide shows that pretty nicely. Um, and so here again, you can see these interactions. So here are my T cells that have been activated. They're now T follicular helper cells. Here are my B cells that have started to be activated. They're moving to the boundary of the T cell zone. Now, oh my gosh, the activated T cells are going to join them. When the activated T cell joins those B cells at that boundary, we get this activation process that I've just shown you. And then we're going to kind of start to move a little bit. I often think of this as moving back into sort of a B cell zone or more of a B cell zone. But we're officially going to go to be in a location called a germinal center now. And so you can see that here. Here's another way you can see that. Activated B cells move to germinal centers. And so you can notice here is my B cell interacting with a T cell. You can see the B cell kind of moving into this location of the germinal center. You can also see up at the top where I can't really reach, the B cell is kind of moving into that big group of yellow B cells called the germinal center. And the germinal center is really the key location where the rest of the steps of B cell activation are happening. If you look at the images on the left, B cells are in blue. So you can kind of see that there is a B cell zone. It's really hard to see. This is pretty washed out. But there's a B cell zone up here, and there's some red T cells down here. So that's kind of trying to show you a B cell zone and a T cell zone and a naive lymph node. And now we're actually going to look at B cells specific to this one antigen. So they're going to be green. You can see that originally they're in the B cell zone. Then they're going to sort of start to like move and interact more with the T cells. And then we're going to get this sort of area of those specific B cells that is this germinal center. So here are the B cells that have moved into the germinal center. Germinal centers are, in fact, really easy to see under a microscope in lymph nodes in a lot of different settings. And so sometimes people might ask a question of a germinal center of kind of where is the germinal center? And there are a couple, and because you could argue that the germinal center didn't exist until the B cell moved there. Like some people are like B cells move to germinal centers, and some people are like B cells move to a place, and that place now gets called germinal center. Like, what was it before the B cell moved there? Is sort of what I'm getting at. And the one key thing about the germinal center, if we want to think where did the B cell move and make a germinal center, is that the germinal center has a lot of follicular dendritic cells. So the germinal centers are happening around the follicular dendritic cell. So here you can see a follicular dendritic cell kind of in this unique location. Remember, our follicular dendritic cell is going to be this big stretchy cell that's going to be in the follicle. And so I often think about this as, at least at the beginning, the B cell, once it's interacted with a T cell, kind of moves to where there's a follicular dendritic cell and like starts a germinal center there. And that germinal center goes on for a while. Actually, one of the papers that I said where we've learned a whole lot about this, we used to think the germinal center went on for, I don't know, a little while. Now we're seeing it probably goes on for at least 6 months. And so, after you get a particular antigen, you probably have a site that's a germinal center that more B cells are going to move into for like 6 months. So the first one made it a germinal center. After that, the rest of them moved to the germinal center. Yep, Brenna.
I just want to make sure. So, originally we were in the B zone and the
Mhm.
T cell zone, and then we went to the border in
Mhm.
between.
Yep.
And then are they going farther in?
No, they're going back to the B cell zone.
Oh, okay.
We're going back into the B cell zone and making a germinal center, or joining a germinal center that's already in progress. And the thing that's key about the germinal center location is that it has follicular dendritic cells. Okay, Brian, do you have a question? That's a great question that people are actually, like I said, that's one of the things we're actually learning a whole bunch about right now. I want you to imagine they're all responding to one antigen. I don't know if that's actually 100% true, but for at least what we're going to talk about right now, you can imagine it with one antigen. Okay, and so there's this other image that I didn't put in here that I kind of wish I had put in here that shows a lymph node with many germinal centers. And so we could maybe imagine that each germinal center is for a different antigen. Yeah, I don't. It's actually one of the most beautiful images. I don't know why I don't have that image in here. But so you could imagine like one lymph node might have like 20 germinal centers. Each of them are where a different antigen is being dealt with. Yes.
Back to like the class switching infinity.
Mm-hmm.
That happen? I don't know because on the pictures.
We are going to get to that still. Nope, great question. That's where we're going. When we think about the germinal center, we can also realize that the germinal center tends to have two different regions that we can see. So here they're less obvious, at least to me right now, in these germinal centers. But let's look at this image up here. I think that that's the most helpful right now. You can see that in this image, the green cells are the B cells. The actually green and blue are B cells. Red are T cells. One of these areas tends to be called the light zone, and one of these areas is called the dark zone. You saw, and they were named the light zone and the dark zone by people who were looking at microscopy images that were much nicer than the ones that I'm showing here at the bottom, as I realize that. Similarly to some stuff you saw with the thymus, actually. So you saw two different zones that in the thymus. They weren't called the light zone and the dark zone, but one was light and one was dark. Why were those light zones and dark zones different in the thymus? Yeah, Brenna. The dark one had more cells present. It was harder for light to get through it. The light zone had fewer cells present. Makes sense, right? Do you remember why the darker zone had more cells present? That was where proliferation was happening. That was where cell division was happening. Guess what happens in the dark zone? We have more B cells in the dark zone, and the reason is because proliferation is largely going to be a key part of the dark zone. In the light zone, the cells are not quite as dense. You can also see we got some T cells in the light zone. And we also, it's not as easy to see here. We also have all of our follicular dendritic cells are in the light zones. So we've got follicular dendritic cells, we got T cells, and we got some B cells in the light zone. And we got a lot of B cells in the dark zone. And when our B cells go to the germinal center, they undergo a process that's known as the germinal center reaction. And the germinal center reaction, if you really think about it, is pretty freaking cool. I don't even like B cells, and it's pretty freaking cool. So here is our B cell that gets activated by a T-dependent antigen. So you can see there's my B cell, T-dependent activation. Now it's going to go into the germinal center. And it's first going to go into the dark zone. So it's going to enter into the germinal center at the dark zone. If it enters in at the dark zone, what can you imagine that cell is going to do based on what we've said so far about the dark zone? What do you think, Brenna? Proliferate. That cell just got a pat on the head. That cell just got told, hey, we need you. Your antigen is here. We need you. So that cell is going to proliferate and make more copies of itself. That cell is also going to differentiate and sort of start becoming a little more effector-like. But that cell is also going to do one other thing. And I will admit, and this sort of gets at your question as well, Juliana, the exact order of the proliferation and the other thing, and are they at exactly the same time or slightly different times, is something people argue about right now. You can imagine you don't want to give someone a lymph node out of your arm and let them like watch this. It's really hard to actually watch and see that timing. So people argue about the exactly at the same time or one right after the other. But the other thing that will happen is that the B cell is going to start getting some mutations. Yeah. It's going to get, the B cell is going to start getting some mutations. The B cell, I know this is going to sound obvious, but there's a reason why I'm saying it like this. The B cell is not like a germ cell, right? It's not like an egg or sperm. It's a body cell. One of the words for body cells is somatic cells. So this is a somatic cell. And it's not just mutating a little bit, it's mutating a lot of it. And so we actually call this process somatic hypermutation. Somatic because it's a body cell, hyper because it's a lot of mutations, and then mutation. And that we can call SHM. So we have this mutation process called SHM, where our B cell is going to pick up some mutations. Our B cell is not picking up mutations across its whole genome, however. Our B cell is picking up mutations at a specific site in the genome at this point. And so that B cell is picking up mutations only at the variable region of the heavy chain and light chain. So I guess two spots of the genome. So heavy chain variable region, light chain variable region. It's the only spot that the B cell is going to pick up mutations. And so you can see an example of that here on the right. Here's my variable region, here's my constant region, and now we've picked up some point mutations as a result of somatic hypermutation. Again, you can see on the left, this is kind of showing you mutation frequency, and you can see all those mutations are really specific for the variable region. Point mutations. So that means that they're one base pair, one letter gets changed to another letter. It's not like a whole big insertion or deletion. It's just you're changing like C to A. It's a point mutation. Okay? If you change the a gene, if you make a mutation in a gene, if you change a G to an A, what might happen if we think about the protein? It'll fold differently, or you'll get a different protein, right? When we have these somatic mutations happening to the gene that encodes the variable region, we end up getting a slightly different variable region for it on the antibody. And so now our antibody that used to have a binding site that looked like this little square thing now has this little curve. And ideally, what we've done is we've gone from an antibody that has low affinity for the antigen, binds antigen weakly, to going to an antibody that binds strongly. So we've gotten higher affinity or better affinity. And so we say what has happened to the protein is affinity maturation. The affinity has improved or gotten better. So our affinity has matured of the protein because we have had somatic hypermutation of the gene. So that's one piece of this. There are so many other things I could say. Yes, David.
Why is it that specific region of the genome that gets mutated and not like anything else?
The big answer for that, and I'm giving you, this is a answer for someone who has taken Molgen, because you've taken Molgen, is that that's where the chromatin is accessible. So you'll imagine that the chromatin is closed everywhere else, opens up right there. That's where and so the mutator enzyme can only access the DNA right there. Yep, Brian.
So the idea that like all of these B cells proliferate and they're all individually mutated.
Exactly. You're getting right there. So so imagine that you were all, remember we had our B cell, it got a pat on the head, it proliferated. So it made 8 progeny. You were the 8 progeny, right? So you were 8 B cells that started out with kind of meh affinity for this antigen. Now you're all going to have this mutator enzyme get turned on, and you're all going to make mutations in your variable region. You're each going to make different mutations. It's a random process. You're each just going to get the mutator enzyme and make some mutations. Okay? Some of you are going to make an amazingly better affinity antibody by chance, right? It's totally a chance thing. Some of you are going to hit the right combo and make an awesome, perfect antibody. But what's going to happen to some others? Brian sort of implied this, but what might happen to some others in this process? Yeah, David.
They don't get as lucky with their mutations.
Okay, but when you say they don't get as lucky with their mutations, what do you mean? Yeah. Or sorry, Brooke. So maybe their affinity isn't better. There's again, we can kind of think of that in 2 ways. One, their affinity goes from 10 to 10. This one went from 10 to 100. One guy could go from 10 to 10, right? What's the other option that could happen? Yeah. It could also lose. It could also basically destroy its antibody. It could go from 10 to 0, right? So you could have some members of the population are going to luck out and get really, really good binding. They're going to improve their antibody. Some are going to basically not change it. Some are going to destroy it, right? What then happens is that B cell goes back to the T cell and the follicular dendritic cell and tests again, tries to bind again. Now let's imagine all 8 of you who have all made different mutations come to the follicular dendritic cell and the T cell who are hanging out in the light zone. Who's going to win? Are all 8 of you going to be able to bind to me, the follicular dendritic cell and the T cell at the same time? No, you're not all going to be able to bind to me, right? Somebody's going to win, somebody's going to lose in terms of getting to bind to me. Who's going to win in terms of getting to bind to me? High affinity ones. The high affinity ones are basically going to outcompete the other ones. They're going to get more signal. They're going to get more pat on the head that said, you did a good job. The other ones are not going to get a signal. They're not going to get a pat on the head that said a good job. And so what's going to happen to them? Because we've talked about lymphocytes and their neediness before. Yeah, David? They're going to die because they don't get a signal. They are basically going to try to test. They're going to try to get a signal. If they don't get a signal, they die. And the only ones that are going to get a signal are going to be the ones who have the highest affinity out of that population. So as a whole, the population is going to move towards higher and higher affinity because only the high affinity ones are going to get a signal, the other ones are going to die. So you can so really this is a matter of natural selection on the cell level in your lymph node that's happening with your B cells. So once your B cells start making a response, you actually get this sort of amplification, this process. Sometimes people ask me, they're like, why does affinity always mature? Why doesn't some B cell destroy its receptor? And it totally does. It's just that after it does, it dies. So you can't really ever measure it. But in fact, some of them will destroy the receptor. So here we got binding. We made some mutations. Here we get nice strong affinity binding. So we get more pat on the head and we divide again. Here we destroyed the binding, so the cell dies. And so that moves the population as a whole from low affinity to high affinity. And so you can see sort of this process happening here. Our cell is going to come into the dark zone, proliferate, and mutate. Then it's going to move into the light zone where it's going to test, can I still bind to the T cell? Can I still bind to the follicular dendritic cell? If so, I'm going to get a signal to live. And in many cases, again, this is sort of stuff we're trying to figure out right now. Sometimes that B cell gets a pat on the head, you did a good job, and then it goes back and mutates again. And sometimes we can see something like 20 rounds where it keeps improving itself over and over. It makes some more progeny, then they get to try again. Then the winner of those progeny gets a signal, and they get to try again. And so you see multiple rounds of the B cell going through the germinal center reaction in order to get really, really high affinity, as opposed to the sort of meh affinity you had at the end, or at the beginning, I mean. Eventually, that B cell will leave the germinal center. We don't know why it thinks it's done. It just leaves eventually. And there it's going to become either an effector cell, which is called a plasma cell. And an effector B cell or a plasma B cell is very much specialized to make extremely high amounts of antibody protein. It's a super, super specialized cell. Or that B cell may become a memory B cell. And so you can see sort of the overall process of the germinal center reaction here. So here is my B cell. It sees antigen, kind of starts to become activated, has to get interact with T cells and some other stimuli. Then it's going to go through its clonal expansion. It's going to go through affinity maturation. So it makes really high affinity antibodies, might become a memory cell, might become a plasma cell to make a lot of antibodies. One other thing happens during this process. And the other thing that happens during this process is that the B cell undergoes isotype switching or class switching. And so now, remember, all of our B cells came out of the bone marrow making IgM and IgD. Now our B cell can switch to making one of the other isotypes, like IgG. And that process only happens when the B cell has gone through the germinal center reaction. Again, where exactly it does that in the process is something that people debate. And so you can sort of see details of this here. Remember, we've got our V, D, and J regions, and we have the IgM constant region pretty close by, and then the IgD constant region pretty close by, and then all the other ones for IgG, IgE, and IgA were far apart, 55 and more kilobases. And I told you that the RNA polymerase will get the VDJ, will get M and D, and then falls off. It can't make it this far. So it never actually makes G, E, or A. And so if our B cell wants to make IgG, IgE, or IgA, we have to make a change to the DNA. So there isn't this super long distance for the RNA polymerase to go anymore. As we talked about previously, different isotypes of antibodies, different classes of antibodies are good at different times. So for example, if you have infection with a parasite, IgE is really useful. So ideally what we'd like to do is we'd like to have a way where we can make the B cell, when it's getting activated, when it's getting turned on in this process, to know, oh, I should be making IgE. We should be able to kind of switch that around, to push that around. And in fact, we can. Because it turns out that which isotype our B cell class switches to is dictated by which cytokine that B cell receives. So remember, I told you that the B cell is going to be receiving a cytokine from the helper T cell, that's signal 3. Well, whatever kind of cytokine the T cells are making will tell the B cell to switch to a different isotype. So you can see, for example, IL-4 tends to induce switching to IgG1, G4, and E, while others will induce switching towards different isotypes. So again, David, just to think about this, part of this is because it's changing which part of the chromatin is accessible. And so we talked about sort of this general problem of this really long DNA and the fact that these other constant regions are so far downstream that we can't transcribe that far. I mentioned that thus we probably have to make a change to the DNA, and we do have to make a change to the DNA. And so what would you guess is the change we have to make to the DNA? Shorten. How do you shorten it? Like if I bought a dress that was for your height, but I put it on me and I had to shorten that dress, how am I going to shorten it? I'm going to cut it, right? I'm going to cut it and then like sew up the end. So what do you think is going to happen to the DNA? We're going to have to cut the DNA again and throw out some DNA. And so that is how we do class switching as well. We use a recombination process. So remember, recombination is when we're cutting and pasting DNA. We're not doing recombination of the V's and the D's and the J's anymore. Now we're doing a recombination of the class switch constant regions, of the isotype constant regions. And so we call this class switch recombination because it's different than VDJ recombination. It does not involve RAG1 and RAG2. And what you will notice is that with each of our constant region exons, we have a little area right before it in the DNA sequence that's listed here as S. It's called a switch site. And so right before, I don't know why they don't put one in front of D. There is actually one in front of D. I don't know why this book doesn't show it. But you can see right before M, there's a switch site. Right before D, there's a switch site. Right before G3, there's a switch site. Right before G1, there's a switch site. So you can see before each constant region gene, there's a switch site in the DNA. And when it's time to class switch, say, again, remember, we always start with IgM. What we actually do is we have a DNA break at the switch sites, and we do a recombination of the switch sites and delete out the intervening DNA. So you can see now we basically have put together the switch sites. Here it was switch mu and switch G1. We basically merged together their switch sites. We cut out all the DNA that was in between, and now we've got the G1 constant region here. And so now it's easy to make an IgG1 antibody. And so you can see, I told you about MDs give everyone apples. Everybody starts out making IgM. You can then you can actually switch from M to D. I don't know why that didn't show us by using the switch site. You could switch to G. You can't go back because you deleted this DNA, but you can actually go forward to E or to A. So you can switch more than once, though I don't know that ever happens super often. And so we get this process of class switch recombination. And so that is sort of the other part of the germinal center reaction. And as you can see here, we can also get differentiation into either our effector or our memory cell. The effector cell in the case of a B cell is called a plasma cell. And so here you can see on the right our B cell becoming either a memory B cell or a plasma cell. And on the left, you can actually see an image of a plasma cell. And what you can notice is in this plasma cell, you've got the nucleus, you've got rough ER, and do you see much else? No. You don't really have much in the way of like a lot of mitochondria. You don't really have some Golgi. Basically, you got rough ER, and you got nucleus, and that's it. And what that allows this cell to do is it allows this cell to make and secrete like grams of protein, but do nothing else. So it can make antibodies and not do anything else. But it basically has thrown out all the other parts of the cell and has become an antibody factory. And so what you can see is that it doesn't have any B cell receptor on its surface anymore. It stopped doing that. It doesn't have any MHC class 2 on its surface. It's not going to grow. It's not going to mutate. It's not going to class switch. It's just going to make all the possible antibodies, unlike other kinds of B cells. And so the effector B cell is this plasma cell, which is basically this really, really, really high level antibody producer. That's all it can do. It has specialized so heavily that it can't do anything else. I have this t-shirt that has like cells of the immune system that are trying to be all cute. Like there's a helper T cell, and the helper T cell says, "I'm helping," in a little bubble next to it. And I think the macrophage says like nom 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 and things like that. The plasma cell on that t-shirt says "I don't care," because it has no receptors. It's not responding to anything. It's just making antibodies and ignoring everything else. Everything I've told you thus far is what we knew about the germinal center reaction largely when I finished undergrad. When I was in grad school, we learned what the mutator enzyme was and some details of how the mutator enzyme worked. I will tell you a bit about that mutator enzyme and some additional details on Friday, so I will see you guys then. Make sure that you have a draft for lab tomorrow.